Hello, I'm David Field. Welcome to this lecture on doing multi-session analysis using FEAT, which is part of the FSL suite of programs. First, let's remind ourselves what we mean by a session. A session is a single continuous time series recorded in the scanner, one set of functional data. So examples of multi-session would be the same participant stays in the scanner for an hour and you do a few different runs of the scanner, some of which are perhaps repetitions of each other. Another example of multi-session would be two sets of data on the same person from 2 different days. What we're going to find out today is basically that higher level or second, third level analysis using FEAT uses exactly the same computational apparatus that were deployed at the first level in order to analyse a single session. That's essentially the message. Now, on the right here we've got what you might be familiar with, a design matrix, but it looks a little bit different to the design matrices you were using at the first level. For example, we no longer have time running from 0 down to the end of the experiment on this vertical dimension. We have instead some numbers that refer to the random effects groupings of participants. And whereas in the first level design matrix a single row in the design matrix was a single time point, single volume or TR, in this design matrix a single row is one participant. Therefore a couple of things are missing from this design matrix that were present in the first level design matrix. First one is you don't see any red bar indicating the duration of the high pass filter, because that's no longer applicable, because you're no longer modelling time, you're modelling participant variation. And the second one is that you don't need to convolve your explanatory variables with any model of the hemodynamic response and its delays and blurring at the second level, because that was already taken into account at the first level. Let's begin with an overview. In the practical session in the workshop that accompanies this lecture, you will cover the 3 most common group analysis or second level analysis scenarios. So the first one is when multiple participants have done the same single session experiment and you want to get the group average activation for one or more of your contrasts of interest. So in a words versus non-words experiment, you could have words versus baseline at the first level, non-words versus baseline at the second level, or the more interesting words minus non-words contrast. And you want to see where on average across the group the brain was active for the words minus non-words contrast. This is formally equivalent to a one sample t-test with a test value of 0, which you may have covered for your behavioural data in SPSS. In the second scenario, multiple participants get scanned more than once, perhaps twice, and you just want to know where in the brain the group average estimate of activation differs between the 2 scanning sessions. For example, you scan someone on a particular task, take them out of the scanner, give them a drug, put them back in later and give them the same task again and see if the drug changed the activation that was produced by the task. And that's equivalent to your SPSS repeated measures t-test. Finally is the independent or between groups t-test, where you get 2 separate groups of participants to perform the 2 experimental conditions. And you're interested in where in the brain activation differs between the 2 groups. So you might be comparing with old people with young people, or you might get one single sample and randomly divide it into 2, and group A get one experimental condition and group B did another experimental condition, and you just did that because it wasn't psychologically practical to give both experimental conditions to the same group of people. So in the lecture today we will remind ourselves of the outputs of the first level analysis, because these outputs are what form the inputs to the second level analysis. And we'll just see how those are combined in performing the second level analysis and what the different options are. So the inputs to the second level analysis are those from 2 or more first level feat runs. In practice, when you're actually doing this in feat, setting up a second level design matrix, one of the things you have to do is select the feat folders from the first level, and it will find the appropriate files it needs within those first level feat folders. So each first level analysis inside those folders provides a COPE image, contrast of parameter estimates image. We talked about words versus non-words on the last slide. Corresponding to that particular contrast of interest is a var COPE image, which is the error variance image from the first level. Remember, that's the variation in the bold signal over time that you were unable to model. So it's your estimate of error. The third image, which is sometimes made use of, is the TDOF image. That's the image that tells you for each voxel what the estimate of the effective independent number of time points at the first level was. Remember how last week we talked about correction of voxel time series for the autocorrelation problem? Well, the TDOF image is the output of that process. So on the next slides, we'll remind ourselves where each of those images come from and what the numeric values at each voxel mean. Okay, on this graph, the blue line is a voxel time course. You're familiar with the way that these tend to zigzag up and down, and that there's sometimes easy to spot the signal in there and sometimes not. On the x-axis we've got time, seems to be in seconds probably in this particular graph. Note the y-axis, all three of these time courses on here have a mean of 0. FSL demeans the data and also the regressors, so that everything will have a mean of 0. Now in green we've got the predicted time course for all the explanatory variables in the model created by multiplying the values of those explanatory variables in the design matrix by the parameter estimates that were estimated through the first level model fitting procedure. So you can see how generally the model is quite good. When the blue line is higher, the model tends to be higher, but it doesn't manage to capture the full amplitude of the changes in the data, so then that results in this red residual line being something other than a flat 0 right across the time course. So, and that's obviously just the that's just the data time course minus the model time course. So we see, if we look at this case here where the data has got quite a high peak in it, that's kind of captured by the model, but the difference between the data and the model is the positive number. So here we've got a positive value of the residual. If we skip forward to this place here around time point 240-ish, here the model is predicting in green a positive deflection upwards in the signal that just didn't happen, and so therefore the difference between the model and the data is in this case negative. We've got a negative value of the residual. So the green line is essentially where those parameter estimates come from, and if this part of the model here and here was one experimental condition, and this part here and here was another, and your contrast of parameter estimates would be the difference between those two values. the two numbers that multiply to make those values. And obviously the residual, the red, is what produces the var cope images. One of the questions that I found myself asking a while back was, why not use only the cope images from the first level to do your second level analysis? The simplest or crudest way of doing a second level analysis would be to average together the values in the cope images from a group of participants who have done the same experiment. You could take it a stage further by looking at the between participant variation in each voxel, as well as the mean activation level. And indeed that is a useful thing to do, as we will see when we move on. But the reason it's useful to actually pass the first level var cope and TDOF images up to the second level is because it avoids throwing away useful information. So I'll give an example of how it might be useful. Imagine two participants who've done your words versus non-words experiment, and they've both got identical values of the cope at the first level. At a particular voxel, they show exactly the same size difference between words and non-words, and it's a positive, interesting difference. So they, using just the cope images at the second level, are going to look exactly the same and contribute exactly the same thing to the second level analysis. However, that ignores the fact that back at the first level, participant 1 had a very consistent pattern of activation, whereas participant 2 could have had a quite inconsistent pattern of activation. So participant 2 sometimes had, on a block by block basis, sometimes the activation for words was much higher than non-words at our voxel of interest. Other times it was only a bit higher, or it was actually lower. Whereas for participant 1, it's a small but consistent, less variable difference. And so actually the first participant kind of provides stronger evidence in favour of the prediction that you're interested in than the second one. And it's useful to take that information about reliability up to the second level, which allows a more sophisticated modelling of the error variance at the second level. So from a practical point of view, the first thing you do when you do a second level analysis is concatenation of images. And as well as being able to do a second level analysis by concatenating images from the first level, you can actually perform a third or higher level analysis by concatenating images from the just from the level below. And one of the examples in the workshop does involve a third level analysis. Now, one of the commonest types of second level analysis is just to take the first level input images from many different participants who've each done the same experiment. So thinking of our words versus non-words example, we could take the words minus non-words contrast image from a series of participants, and we need to basically concatenate or stitch them together to form a 4D image file. Now the fourth dimension in this image file is different from how it was at the first level. At the first level, of course, it was time, and in the fourth level it's participant. And if you can manage to get your head around that, the fact that you're modelling participant courses rather than time courses in the second and higher level analysis, then you can understand what I said on the first slide, that we basically can then apply exactly the same computational apparatus that we employed at the first level at the second level. Of course, the particular order within the participant course is arbitrary and up to the experimenter, because it will just depend what order the experimenter does the concatenation in. But of course, that order that you've set up when you do the concatenation has to be then respected when you set up the regressors, as we will see. Now, as well as performing the concatenation on the cope images, you also perform the concatenation on the TDOF and Varcope images from the first level. As I said earlier, second level analysis basically works in the same way as first level analysis. You need to set up a design matrix that embodies your beliefs about likely systematic sources of variation across your participant dimension. And each belief is in effect an explanatory variable in the design matrix. Just like in the first level analysis, we're using a mass univariate approach. The design matrix is fitted separately at each voxel location, and that fitting will result in parameter estimates for each of the explanatory variables at each voxel, just like it did at first level. And then once again, you might, if you have more than one explanatory variable at the second level, perform 1 - 1 contrasts at the second level, or you might just produce versus baseline contrasts at the second level, producing second level cope images. Okay, so let's take a look at what the design matrix will look like for a independent samples or unpaired t-test. This is the situation where you've got two groups of participants and you want to test the hypothesis that their patterns of activation come from two different hypothetical populations of activation. So I'll give you a couple of examples. It could be that group 1 are elderly and group 2 are not, or it could be that groups 1 and 2 are in fact from the same population of young people, but then they experience something a bit different in the scanner. It could be that you just give slightly different versions of the task to groups 1 and 2, and that you couldn't do a type of experimental design where every participant experiences both tasks, because that would have compromised the psychological validity of the task. So these numbers down the left hand side, they represent the random effects groupings of the two sets of participants. So this embodies our belief that this group here, group 1, all these rows in the design matrix are images that have come from participants that are part of population A, whereas group 2 are images that have been randomly selected from population B. So now we come to look at the actual explanatory variables. If we look at the first explanatory variable, it has a positive value for the first group of participants, and the value for the second group of participants is 0. Now the second explanatory variable is the reverse of that. It has a value of 1 for the second group of participants, and it has a value of 0 for the first group. Now the fact that there are values of 0 in there is important, because it means that whatever the parameter estimate turns out to be, that you multiply this explanatory variable by the resulting model fit value on that explanatory variable for group 1 in this case is always going to be 0, isn't it? Because 0 times anything is 0. Remember, we saw the importance of this in the week where we used the interactive spreadsheet to look at how we fit individual time courses. Any non-zero parameter estimate on this regressor is modelling the fact that the second sample have got, on average, across the participants, a parameter estimate from the first level greater than 0 at that particular voxel time course. So let's talk about the meaning of these contrasts now. We've set up the 1 - 1 contrast, group 1 minus group 2, or group A minus group B if you like, and the reverse, group B minus group A. So what those contrasts are asking is in effect the question, at a particular XYZ voxel location, is sample 1 more active than sample 2? And for this second contrast, it's asking, at a particular voxel location, is sample 2 more active on average than sample 1? So if we had a voxel where both populations activate about equally, then of course the result will be 0 in the cope. Okay, this design matrix is actually the simplest one, and people when they're doing these kind of lectures, they often show this one first because it's the simplest and it's the most commonly used. But it's so simple that it's not obvious what's actually going on, what it represents, which is why I showed you the independent samples design matrix first. So this is a single group average design matrix, or one sample t-test. Basically for the case where you've got a group of participants who've all done exactly the same thing in the scanner. So let's say they've all done the same auditory and visual experiment, and you just want to find out where on average across the participants there is activation for visual, and where on average there's activation for the auditory stimulus, and also where on average there is differences in activation levels between those two types of stimuli. So in this design matrix, what we have down the left is once again our belief about the random effects groupings of the participants, and the fact that those are all ones just says we think we got all these people randomly from the same population. Now, a single explanatory variable in this design matrix just has a positive value for every participant. So therefore there aren't any participants being excluded here from this explanatory variable as they were on the last slide, because there are no values of 0 in the explanatory variable. This positive number will just get multiplied by whatever parameter estimate is the best one to fit the combined data of the participants, and of course the best model is basically going to be the mean at each voxel location across the participants. So this will in effect calculate the mean across the participants at each voxel. Now, I mentioned earlier that in an auditory and visual condition you could use this design matrix to find the activation voxels for auditory, find the activation voxels for visual, and find the activation voxels where auditory and visual were different. But look, we don't have contrasts that do that at the bottom here, do we? There's only one contrast. And the reason for this is that this design matrix at the second level can be applied to as many or as few of the first level contrasts that you processed as you want. So you can tick in feet boxes to apply this to all of the contrasts that will run at the first level. So if the first level feet folders contain contrasts for visual, for auditory, and for auditory minus visual, and for visual minus auditory, and then you set up this second level design matrix, it would actually produce 4 second level contrasts of parameter estimates, one for each of the first level ones, or you could choose to select a subset of the first level ones if you want. So in effect, this design matrix is testing the null hypothesis at a particular voxel that the activation on average at that voxel is 0. These are just the labels that I forgot to show as I was talking about all that. So that just shows you that once again the model is just one population of schools, and it tells us that all the participants here are going to be weighted equally because they've all got a value of 1. And in effect, this contrast just takes the parameter estimate 1 image and copies it to produce cope 1. Let's take a look at finding the voxels that are active at the second level. Now we know that a parameter estimate will be produced at each voxel for each explanatory variable in the design matrix. Now, just like at the first level, those parameter estimates have in effect arbitrary units, and you need to standardize them, and just like at the first level, they're standardized in relation to the unmodeled or residual variation by dividing them by some estimate of the residual variation. Here's where second level analysis gets a little bit more complicated. There are choices to be made here in relation to how we actually estimate the residual variation, and we'll take a look at that now. Let's take a look at a participant data series at a second level voxel. So here in this graph, participant 1 is on this row here, and the x-axis is the effect size. Okay, and we're going to add more participants on the other rows. So this person has this effect size, and they also have associated with that effect size a value from the VARCO image, which is the standard deviation of the residuals from the time course model at the first level. Now, as well as our first participant, let's say we've also got these other rows. So participant 2 is shown on the second row here. 3, 4, 5 and 6 are also shown on their own individual rows. So each participant showed a different amount of activation at a particular voxel, with the most activation being shown by participant 6, and all of them had activation that was greater than 0, so it looks like we are going to find something at this voxel. The mean of the effect size is now shown here by the dotted red line that's appeared vertically. Now remember, as well as the mean, we've also got our estimate of the unmodeled variation for each participant from the first level, which are now being shown by those little red hats, red Gaussian distributions on top of the individual participant means from the first level. So we can use that information to perform what's called a fixed effects analysis at one voxel, which is one way of thinking about your error variance. And in a fixed effects analysis, the variance or error term is the mean of the separate within subject variances. So these are all shown as actually. So to recap, fixed effects analysis is conceptually pretty simple. You average the different effect size estimates from the different participants, and you also average the individual error variance estimates from the different participants to get your error term for running your t-test. This was what was done for group analysis purposes in many of the early FMRI publications. It's actually equivalent to treating all your participants as if they were one participant. So given that individual participant time series at the first level have been demeaned anyway, you could then actually concatenate them, put them all together into one really long time series, and set up one first level design matrix, albeit a very big one, that modelled that variation in your sort of arbitrarily created very long time series. And that would actually be formally equivalent to a fixed effects analysis performed at the second level. Now, fixed effects analysis has fallen out of favour with journal article reviewers and editors, and it's difficult to publish those now when they're used to perform group analysis. And that's because, from a statistician's point of view, what you're asking is, what is the best estimate you've got of the mean activation at each voxel for a particular contrast for that exact group of participants who perform the experiment. You're ignoring variation between participants. Your participants were some sort of sample, hopefully a random one, from a population, and you want to generalize to that population. So you need to take into account the variation that is present in your data due to sampling from that population. You have to do that if you want to be able to make the inference that your group results reflect something about the population that you took your group from. We'll see how to do that in the next few slides. However, fixed effects analysis is still commonly used, and we'll do this in the workshop. It's used for the situation where one participant has performed multiple sessions of the same experiment. You just want to average across the sessions. For example, if you're doing a task where the participant has to fixate throughout the whole task, it's quite hard to fixate well for more than about 5 minutes. So you might want to let them do it for 5 minutes, give them a break where they can move their eyes around, and then just do some more trials of the same thing with them fixating again. And in that case, it's probably a reasonable assumption, given that you haven't even taken them out of the scanner, that variation between the scan sessions is fairly minimal. Maybe if it was on different days, it would no longer be a reasonable assumption, and it'd be better to model this factor as a random effects factor. So let's take a look at the alternative to fixed effects analysis, which is called random effects analysis. This can be used to ask whether we believe, based on the data in our sample, the actual population at large activates on average for the cope that we are interested in. So the difference in this diagram compared to the earlier ones I was showing you is this big Gaussian, which represents the between participant variation. Now all it is just the standard deviation of the individual participant effect sizes or cope magnitudes, okay? And that can be contrasted with these within individual participant variances that I showed you earlier. So in the random effects analysis, we're going to use the between participant standard deviation to calculate our error term, whereas in the fixed effects analysis we were only using the within subject variance. If we do something called a mixed effects analysis, we're using both sources of variance, which is the optimum thing to do, because then you're taking into account which individual participants are giving more reliable estimates as indexed by their within participant variation, but you're also taking into account the between participant variation, which is the most important kind of variation. Now, as you might sort of gather by looking at that diagram, the error term produced by averaging the 6 small distributions, as in a fixed effects analysis, is usually going to be smaller than using the between subject variance as the error term, which is perhaps one of the reasons that early FMRI studies tended to go for the fixed effects analysis, because then they could use smaller samples. And that's because, therefore, fixed effects analysis is more sensitive to activation, will give you bigger t-values for the same number of participants and the same amount of time in the scanner compared to random effects. But the big downside is this lack of ability to generalize your results to the population. So let's take a look at the various higher level analysis options available in FSL, in particular the mixed effects analysis. But if you want the higher level error term to be made up only of the between participant variates without taking into account the lower level estimates of variance from modelling the time courses, then select OLS, ordinary least squares, in feet. The advantage of that is that it runs faster than the others. So if you're just trying out a lot of different analysis to see what works best, you're varying other things, then you might want to try this method just because it's quick. But if you want FSL to also make use of the VARCOPE and effective degree of freedom images from level 1, choose Flame 1. That makes use of first level fixed effects variance as well as the random effect variance that we saw on the previous slides in constructing the error term. Degrees of freedom are also carried forward from level 1. FSL say that group activation could be more or less than using ordinary least squares. It depends. Either way, it should be more accurate. Another option provided in feet is something called outlier deweighting. This is a way of reducing further the between participant error term when some of the participants are a bit different from the others, or what you might call outliers. As well as reducing the size of the error term, it reduces the impact of those outliers on the mean, so they have less weighting than the cases that seem to be more typical in determining the mean. And it works by assuming the sample is drawn from two populations, a typical one and an outlier population. And for each participant, it's going to estimate the probability that that data point is from the outlier population and de-weight it if it thinks that it is. Note that it does this on a voxel-wise basis, so a particular participant could be judged to be an outlier in one group of voxels in one particular part of the brain, but not in others. Let's do a recap of higher level design matrices. Remember that in a first level design matrix, time runs from top to bottom, whereas in a higher level design matrix, each participant now has one row, and the actual top to bottom ordering has no influence on the model fit, but experimenters tend to group together participants who've done particular experimental conditions because it makes it easier to interpret the design matrices. That first column is now a number that specifies the group membership, and it's going to be 1 for all participants if they were all sampled from one population and all did the same experiment. The other columns are the explanatory variables, which embody your beliefs or predictions about what systematic sources of activation there are going to be in certain parts of the brain. Just like with the first level, at the bottom we specify a set of contrasts. And an important thing is, given that this higher level analysis is sitting on top of the first level analysis, you get to choose which first level contrasts have a particular design matrix applied to them. By default, your second level design matrix will be repeated for every first level contrast that you've specified, and therefore you're going to get one second level cope for every first level cope just by setting up one second level design matrix. In this couple of slides we're just going to revisit the meanings of contrasts to make them absolutely clear for the unpaired or independent samples t-test, perhaps using the example of patients versus controls. So contrast 1 subtracts the image PE2 from the image PE1 to produce image COPE1, and in this COPE1 image, voxels with positive values are more active in controls than in patients. In in contrast 2, the subtraction is done the other way around. We subtract image PE1 from image PE2 to produce COPE2, in which voxels with positive values are more active in patients than in controls. FSL uses two different between participant error terms, one for each grouping, the grouping being this on the left. Therefore it's okay to have different sized participant groups. In this case we've got less people in 2 than in 1, and it's okay if the two groups have different variants. Don't need to worry about that. Now this design matrix is a particularly interesting and useful one. I've made good use of this over the years. It's the single group average or one sample t-test design matrix with the addition of a covariate. A covariate where you've got a value that varies between participants. So in this case the covariate that's being modelled is the reaction time to the task that they were performing in the scanner. So here our group mean will give average activation for the task across participants, but this contrast on the reaction time is going to give us activation for, or rather it will highlight voxels that are particularly active in people who are slower to respond in the task. And if we were to replace that 1 on the second column with a -1, or add a third contrast with that -1, then it would show us voxels which are more active in people who are faster on the task. And how this works is that the explanatory variable has higher values for people who are slower in the task and lower values for people who are faster in the task. And this explanatory variable has been de-meaned or mean-centered, so it will have a mean of 0. And in order for a voxel to get a parameter estimate much more than 0 for this explanatory variable, the pattern across participants of activation will have to be such that the activation is more in the voxel for this person, less for this person, and somewhere in the middle for the others. If a voxel participant course has that pattern, then this will be the explanatory variable that gets a high parameter estimate in that case. And of course this is really useful. You don't just have to do reaction times, you can put in other parameters of interest as well. Bear in mind that you should always consider these extra covariate regressors in the context of the actual task that was being performed in the scanner. The meaning is going to be different if it was a resting state scan compared to a Stroop task with incongruent trials. That just reiterates what I said earlier, that contrast 2 is going to locate voxels that are relatively more active in people with slow reaction time and less active in people with fast reaction time, and that you'd use a contrast of 0 -1 to locate the brain regions that are more active in people with quick reactions and less active in those with slow reactions. Now let's look at the paired samples t-test and how that is implemented in FSL. So in this design you've scanned the same participants twice. For example, you've scanned some people on a memory performance paradigm, both with and without a drug. Now you want to be able to calculate the difference for each person separately between the time 1 scan and the time 2 scan at each voxel, because you want to analyse the difference scores, not the raw scores. So the variance in the data due to differences in mean activation level between participants is not relevant to you here in the same way as it is if you are doing the independent groups design, where you'd have to take that into account. You were just interested in time 1 versus time 2 within participants, if you like. This is dealt with in feet by passing the data up to a second level with those between subject differences still present, but then modelling it out or removing it using what you might call nuisance regressors. Let's take a look at what the design matrix for a paired samples t-test looks like in FSL. The first thing to note is that all the participants have been assigned to the same random effects grouping. This is one group of participants as far as we're concerned. Now we can see there's a lot more actual explanatory variables in this design matrix than other ones that we've been looking at so far today. And the ones in the top half of the design matrix are the first level contrasts of parameter estimates from the drug condition, and the ones in the bottom half of the design matrix are the ones from the no drug condition. This explanatory variable here is the one we're really interested in and the one we want to run our contrasts on. If you see, it's got a high value for all of the drug condition scans, and it's got a low value, negative value, in fact, for all of the no drug condition scans. And so this contrast here of 1, which is run on this regressor, that the regressor itself actually embodies a 1 minus 1 difference anyway, so then you just need to run a contrast of 1 on it rather than 1 minus anything. And that contrast of 1 will pull out for you voxels in the image that have higher values when the higher activation when the participants were taking the drug and doing the memory test than when they were just doing the memory test having not taken the drug. And if you were to run the flip side of that, the -1 contrast, that would find voxels that were more active during the memory test when people were not taking the drug. Now, what's the purpose exactly of all these other regressors? If we take a look at this regressor here, it has a value of 0 most of the time, but occasionally, just twice in fact, it has a positive value. And the 2 occasions on which it has a positive value correspond to the 2 rows in the design matrix where the data comes from participant 1. And the one next to it has positive values for the 2 rows in the design matrix where the data comes from participant 2. Once when participant 2 did the task without the drug, and once for when participant 2 did the task with the drug. And therefore, these explanatory variables on the right hand side, each of them will model out any variation in overall image intensity due to between subject differences. So if it were to happen that participant 1 here happened to generally have much higher image intensity values than all the other participants, you'd get a large positive parameter estimate there. And let's say participant 3 tends to have about the mean of the series for their image intensity values. You basically get parameter estimates of about 0 there. Participant 3, well, their images are a bit darker than other people's, so their overall image intensity values are lower in general, so then we get negative parameter estimates for that particular explanatory variable in order to model that out. So we're not really interested in looking at the results of these explanatory variables. They're just there to model out variance that we don't want to include in our error term. That's a common theme with many of the design matrices we've been talking about, that some of the regressors are just there to reduce our error variance. Finally, something I haven't talked about so far today for group analysis, but which is actually a really important thing to bear in mind, is that any higher level analysis is only as good as the registration of individual participants to whatever common space or template image you are using. you are assuming when you do group analysis that voxel x10, y30, z minus 20, or whatever voxel you're interested in, corresponds to the same bit of anatomy in all of your different participants. And that's a pretty big assumption, and it's only going to be even approximately true in the cases where your registration is performed correctly. If registration is not good, the anatomical correspondence between two participants is poor, and you can't really meaningfully then assess the functional correspondence between those participants. Now note that registration algorithms tend to go wrong more often when participant brains are more different from the sort of template. So if the patient groups are elderly, they go wrong more often. You need to repeat the registration with slightly different parameters usually when it goes wrong until it produces a solution that at least visually makes sense. So do check your registration results at the individual level. Don't just run a whole batch file that just runs a whole set of first level analysis and then does the second level analysis for you, and then you're only interested in the second level analysis because that's the one you want to publish, so you ignore your first level results. That's a disastrous thing to do. Make sure if you're in that situation where you're batch processing data that you at least look at the registration results of the individuals, and also I would say look at their motion plots as well to see which ones may have moved excessively. That's the end of the lecture.